Good afternoon. Welcome to the Schmaltz meeting and the membrane protein Paris meeting. I would like to introduce our first chair, Johanna, who has been a presenter on the Schmaltz Network conferences before. She's going to be chairing our first session. So please kick it off if you could, Johanna, and thanks everybody for joining us today.
Sure. All right. Welcome everyone to this first session. I am going to introduce Claire Naylor as our first speaker. So she's from Quantifoil and she's going to talk about HexAuFoil grids, which I know everyone who does Cryo-EM is really excited about. And then after Claire, we have Reid from Domainex talking about biopolymer encapsulated nanodiscs. Which promises to be very exciting. So if I could ask you to write any questions that you have in the chat, and I can read those out later, or if you'd like to ask your question yourself, if you could use the sort of raise hand emoji reaction thing, and then I can see you wanting to ask a question. So Claire, if you could share your screen.
Sure. There.
Perfect.
Move into slideshow. Hopefully everybody can see that okay?
Yes, we can.
Good, brilliant. Thank you very much for inviting me, and it's a delight to be the very first speaker of this Schmaltz membrane protein meeting. I'm very honoured that you've allowed me to speak. So today I'm going to quickly run through a little intro about the HexAuFoil grids, but first, perhaps appropriately because I'm the first speaker, a little bit of history. So when we started doing Cryo-EM, we had just to remind everybody, the traditional CEM grid was in use was a metal mesh with a 10 nanometer thick carbon film. And the Quantifoil, the original thing about Quantifoil, why we're called Quantifoil, is that we were able to produce holes with predetermined size and arrangement in a wide variety of patterns, and that was really what was used from when they were invented in 1999 for at least 15 years and was the standard grid type. The problem with these, as was shown by Chris Russo from LMB, is that as you freeze things, with the differential effects, freezing effects of carbon and metal, and also water and metal and carbon, you get strain introduced into the system, which is released when you locally heat the sample. And this is a major contributor to beam-induced motion, which is a major part of the motion that you see when you first expose your sample to radiation. And so you lose your least radiation damaged data due to the effects of this of the differential motion between your carbon film and your metal grid. As I think everybody knows, about 10 years ago now, Chris initially came up with one solution to this problem, which was that you simply made the film out of gold, and that was commercialized by us as Ultrafoil, and that eliminates this differential contraction of carbon and metal. And has been shown quite extensively, perhaps best by Radodenev about 3 or 4 years ago, that you do gain about half an angstrom of resolution just from this simple, not so simple, moving from carbon film to gold film. Okay, so solve the problem. So now we've addressed the motion of the samples within the plane of the grid, because you don't get carbon film crinkling due to a differential contraction of the metal and the gold of the carbon. However, the ice also expands differently from the metal, and you get a compressive strain that causes the grids to kind of buckle upwards. You can think of it as a bit like kind of a trampoline. When you press weight on, it goes up and down, and you hit it with the energy, and you get this strain released, and you get a motion almost kind of perpendicular to the grid. Chris was able to show that that this buckling occurs if your grid is more than, if the width of your grid is more than 11 times the thickness of the ice that you want to get. So 30 to 40 nanometers thickness of ice, you need holes that must be smaller than about 0.3 micrometer. And he showed this very nicely with some tilted data where you get very much more movement in the perpendicular to the grid. So this is a tilted and this is planar as you go up in hull size. Okay, so there's a small problem with that, which is when you make a HexAuFoil or an ultrafoil, the process is done by floating a pattern film onto a grid that's brought in. Now, that leads to a smallest hull size of 0.6 micrometer and a variation in hull size. So this is whenever you're using any carbon grid or ultrafoil of plus or minus 0.2 micrometer. So we're trying to get to a hole of 0.3 micrometer, and our variation in hull size is almost that big anyway. So Chris didn't give up. He invented a completely new way to make grids. And this really is going to be the heart of my talk here, this completely new way to make grids so that we can get to this 0.3 micrometer hole size involves now making grids as a single unit. We start by doing a process of lithography from a pristine wafer, a lithography process and cryo-evaporation of gold to form at a wafer scale your film. But the film is now patterned so that it's got you've got 1,000 grids, grids worth of film patterned with the shape of the individual grids on it. And then we do another piece of lithography onto that film to make the grid. And then we release those grids from that wafer and pack them, and this becomes more. But the beauty of this cryolithography, then lithography, gives us really great control over what your grid looks like. We can go down to holes as small as were needed, but even more important, our variation in hole size now is plus or minus 10 nanometer. So every grid now is the same in a way that it fundamentally wasn't, that can give you consistency. And that's really the most important thing I think about these grids, as I'm going to show you, is that they're consistent in a way that traditional grids just haven't been. Okay, so there you go, just to show you what they look like. You've got 50 micrometer hexagonal squares with 300 nanometer holes. You have 9 million holes on a HexAuFoil compared to about 850,000 on a standard R2, R1.2, 1.3. You do have to use them at high mag, and we have introduced a bunch of fiducials to make it easier for you to see them at high magnification. When you're using them, you need to glow discharge them longer, you need to blot them much larger and longer and harder, and data collection at 165,000, if you're going to get the advantages of these high density holes, you really need to make sure your eucentric height and your image shift is absolutely spot on. It needs to be done just before you do the data collection, and then you will see great consistency. And this is just an example where if you just leave the same image shift correction after the last session of the microscope, you kind of miss the hole at the edge of the aphis region because you're at 145 nanometer. But if you just recalibrate, you get most of every hole. And just to say, you can use it on glaciers, but because of the beam, if you don't have fringe-free imaging, you do have to skip, but you can use them. Okay, so I've just given myself just about enough time. There you go. If you've got a hole, you can hardly see the ice is there because it's so flat. You've got a 35 nanometer ice plus or minus 6.5 nanometers across the whole grid. So you can really use every hole on your grid. And this returns, on the left, you have HexAuFoil, on the right, you have ultrafoil. It means that you have very, very consistent. So the relative ice thickness here is going 1 to 1.4 compared with an ultrafoil, which is flatter than a carbon, of 0.5 to 2. So, and you can see that's reflected in the CTF and your average defocus. Somebody said to me, this almost looks like you've just drawn an array here, but I promise you it's real data. And what that pays forward into then is that your HexAuFoil, you get a little bit of extra resolution. This is 2.57 to 2.35, but the maps are less noisy. So you will see more features because you've got less background noise. So even though some of the sub-angstrom motion stuff isn't very relevant at mid resolutions, this less noisiness is going to make a big difference to your life, and the consistency. It makes processing and things much easier. Okay, that's just to prove that, yeah, even if somebody else does it, particle tracking is the same. You know, you go from ultrafoil where you can see motion to particle tracking, all the motion in your particles eliminated. I'm not going to go through zeros unless somebody asks me. There we go. This is my favorite quote at the moment for what's good about pleasant experiments with the HexAuFoils. For a bit of microscope presets tuning, the grids yields crisp maps with the lowest ever B factors on the target protein. For small molecule brand structures, the HexAuFoils will be my go-to grids. And I just want to say thank you to everybody at Quantifoil that helped with development, everybody at SPT, of course, Chris and Katerina at the LMB, but also all the people whose data I've just shot through there that shows everybody, Peter Harrison at EBIC, Felix Weiss at IBS, Yehuda and Sebastian, people at USCD, so many people who have tested and been kind enough to share the HexAuFoil images that they and data that they've had of the early grids. Okay, and yeah, we've got a new building coming, so your lead times for everything else should get lower. Thank you very much. I think I just about managed it.
Thanks very much, Claire. I think this is super exciting. I haven't yet used HexAuFoil grids, but it's definitely on my list of grids to try. Maybe I'll kick off with a question while people are coming up with their own. So I was wondering, clearly the HexAuFoil grids are going to mostly address the problem of motion, right?
Yeah.
So I was wondering, are there sort of particular, in addition to that, are there particular samples, like is it better for smaller proteins versus larger proteins? Does it have a particular effect on the ice thickness as well that you don't see in other?
Okay. So what I would say, first of all, is yes, it's an effect on motion, but I would say that almost more important is this thing I said about how the grids are now more consistent, so that you get much more straightforward processing, and actually it seems to be paying forward to really, really clean maps. So it's almost, you know, if you think about, we're all looking for consistent reproducible sample prep, yet the thing that we're doing the sample prep on until now has had quite significant variability, and we've eliminated that. So that's got to give you more reproducible sample prep. But yeah, at the moment the holes are quite small. We are seeing the best, the people get most excited about it tend to be small complexes, small membrane proteins. But we have the whistle-stop tour there, the images I showed you comparing HexAuFoil and ultrafoil maps, you could see that was quite a large membrane protein actually. So we have, people have used them across the spectrum.
Thank you.
Okay, thank you. Thank you for having me.
Absolutely. Well, I don't see questions at the moment, but if you want to write your questions down in the chat,
Yeah.
I think Claire will be available to answer them in the chat as well.
Yes, and I'll be around tomorrow at the main meeting. So if anybody wanted to catch me there, very welcome to ask questions there as well.
Excellent. Thank you so much. We are running a little bit ahead of schedule. I don't know, Jim, are you ready to put your slides up?
Yes, I can hope you can see and hear me okay. I'll just screen. Okay.
So just to introduce Jim here before he starts, he's a senior principal scientist at Domainex, and his talk is going to be about the fragment screening of adenosine alpha receptors using polymer-encapsulated nanodiscs. And as somebody who's used a lot of nanodiscs myself and is always looking for the latest and greatest ways of using those and optimizing their use, I'm very excited to hear this talk. Take it away.
Okay, thank you. Well, thank you, Evelyn, for the organisers for the invitation and for allowing me to talk about some of the newer assay technology we've had in-house and how we've managed to use this to set up a fragment screen for GPCR in SMAP-like molecules. Okay, so just a quick introduction to who we are. So Domainex is a CRO based on 2 sites close to Cambridge in the UK. We have a global client base. We have a fully integrated drug discovery pipeline covering through from gene, protein production, assay development, medicinal chemistry through to lead candidate generation. So we're primarily based in the small molecule drug discovery area. And so I want to talk to you today about our fragment screening setup. So we have an in-house library of over 1,000 different fragments. And just as a description of what fragment screening is. So when we're talking about, I'll just put my laser pointer on. When we're talking about fragments, what we're talking about small chemical entities, usually less than 300 Daltons in size, low complexity and structure. This, compared to traditional high-throughput screening of mature drug molecules, allows us to very quickly identify small pockets of binding on your molecule using a very small number of compounds. So typically a few thousand compounds will cover a very broad chemical space. So these are fragments that can then be used as starting points to expand these into larger molecules with higher affinities, higher specificities. So the disadvantage of this is these fragments, because of the nature, tend to be very low affinity, which makes them very difficult to detect by many assay techniques. So cell-based assays would be quite difficult to find something of this low affinity. So that requires a very sensitive binding assay, and it needs very high quality protein reagents to do this, which in the case of membrane proteins is going to be particularly challenging on both counts. We have in-house done fragment screening on quite a wide range of target classes, particularly soluble targets. But we wanted to set up a case study to show that this is possible to do as well with membrane-bound targets too. So we chose the A2A receptor as a candidate for a case study. So apart from being a clinically relevant Class A GPCR, it's also very extensively studied. So plenty of structures there with several very well validated ligands from very high potent nanomolar affinities to micromolar affinities. And particularly important in this case, some of the known antagonists like caffeine and theophylline are already fragment-like molecules, so very low affinity, very low, small sizes. So we have something we can use to set up an assay. So to do this, we wanted to try out the known detergent purification methods, so several different conditions known in literature. So we're using LMNG to purify this into detergent micelles, but also to do this in SMALPs as well. So since there are now over 30 different copolymers commercially available, we need to do a solubilization screen to identify which is the optimum polymer to go forward for purification first. So we do this in a 96-well based format. Typically, we would do this sort of thing using a membrane fraction first, but in this case, the A2 receptor was expressed well enough that we could do this directly from insect cells. So it was expressed in insect cells in the back of our expression system. It's solubilized with a range of different polymers or detergents, incubated, and then insoluble impurities removed, and then Western blotted to look to see which is giving us the highest solubilization efficiency. So we're getting so the cycloalkane amphipoles, the ultrasolid amphipoles are working quite well. Several of the SMAs are working and ASTIs are working. But just because it can solubilize doesn't necessarily mean it's feasible to purify that as well. So we followed up with an affinity capture on affinity resin. So in this case, it was via his tag, so we're using talon resin. So we incubate overnight with affinity resin, elute, and then look on Western blot to find which are capturing well. And the standout ones in this case were the ultrasolid amphipoles. Using this assay setup, we're then able to further optimize this with incubation time, the amount of polymer, salt concentrations, buffer compositions to get the best conditions. So skipping forward to actually purifying the protein. So we purified both with LMNG and with the amphipole. In both cases, we were able to get equivalent levels of purity and yield from size exclusion traces. We can see, as we'd expect with the amphipole, we have a much larger size distribution, so around about 400 kilodaltons, which is what we expect for a nanodisc of the broader size distribution, because the nanodiscs we suspect are going to be a more or less uniform size compared to the polymers, sorry, the detergents. But in both cases, DLS analysis shows that we have a single monodispersed species. So the polydispersity on these is both 20% or less, so it's a nice monodispersed species. And we then took this forward to look at whether we could see drug binding. So our first assay that we used to look at compound binding was NanoDSF. This is a thermal denaturation using intrinsic tryptophan fluorescence. So in the case of the detergent solubilized protein, we're getting an inflection point, a TM of about 53 degrees. And in the presence of caffeine and theophylline, we're seeing about 2 to 3 degree shift, increasing stability, and a very high potency ZM compound. So this gives a much larger shift up to about 65 degrees. And we're seeing the same thing with the polymer solubilized protein. So in both cases, we're seeing the same sort of shifts going on. Arguably, we can say that the polymer purified protein is actually giving a nicer, cleaner signal compared to the detergent. There's less interference from the detergent prep, but both are behaving the same. This particular assay doesn't give us any affinity data. It just tells us whether or not we are seeing protein compound binding. So we took on another assay. So this is a fairly new assay that we have in-house. It's an upgraded method from Nanotempera on the Dianthus platform. So spectral shift is looking at the fluorescence of a label attached to your protein. And the dye's chemical environment changes very, very slightly when you add a compound, either red or blue shift. And they have very sensitive optics to plot that difference between the two wavelengths, 670 and 650, when you add a compound. So these very slight changes in the environment around the dye can be caused by either the close proximity of the compound to the dye or through conformational changes induced by ligand binding. So looking at the A2A, when we tried applying this to the detergent-based purification, we couldn't see any dose response curve for binding to this receptor using either the very high affinity ZM compound or the low affinity caffeine or theophylline. This could be due to interaction between the detergent and the fluorescent dye. So apparently some of these dyes do get tangled up in detergent micelles, and that can cause effects on the outcome of the assay. But in the polymer purified protein, we have a very different story. So we can see very clear dose response curves with KDs of the expected values for caffeine and theophylline. They are micromolar affinity compounds. We have another medium intensity affinity compound, DPCPX, which is binding at high nanomolar, and the ZM compound. So the hill slope on this particular curve is very steep, because that's because we've actually hit the tight binding limit of this assay at very low nanomolar concentrations. So this means that in this particular case, the smelt-like molecules are giving a much better readout and actually enabling us to use this assay, which we wouldn't have been able to do with the detergent-based purification. And the full dose response curves for low affinity ligands means we can take this forward for fragment screening, because we can get that detection of very low affinity compounds. So that's what we did next. So we took a library of around 960 different fragments, screened at 250 micromolar against the A2A in Nanodiscs. We used theophylline as a control, which is in the gold here. The software for this particular equipment very handily shows up inconclusive and autofluorescent molecules. So these are due to aggregation or due to autofluorescence of the compound. But the ones we're interested in are in purple. So these are flagged as potential hits. And we had a very respectable 9% hit rate using a user surprise threshold. We took some of those forwards, did an 8-point curve to get KD values for this. And we have a range of different molecules which are confirming as binding with micromolar affinities and good ligand efficiencies as well. So anything above 0.3 is a good ligand efficiency for this kind of molecule. And interestingly as well, one of the compounds from this pilot screen, 9230, is a very close analog of caffeine and theophylline, so further validating that we are actually pulling out valid hits from this kind of assay. So this now means we have an assay which we can take forwards for a medicinal chemistry drug discovery pipeline. And as far as we're aware, this is the first time this has been described for fragment screen using polymer nanodiscs style molecules. Okay, so just as a thank you to all the people who've been working on this in our team. So we have a few people here. So I'm based in the protein sciences division, and all the assay work was done with for the assay biology group, and the chemists are providing all the compounds for the fragment screening. Okay, thank you very much.
Thanks very much for that interesting talk, Jim. So if you have questions, please ask them in the chat, turn your video on and raise your hand, or use the react button to sort of virtually raise your hand. I'm going to start off with a question about pH restrictions, because some of these SMOPs have very specific pHs in which they work at. Was that something that you had to consider during your fragment screening, to only work in a specific pH range, or that wasn't really an issue?
So it's we can change the pH a little bit. So this spectral shift assay is a full solution-based assay. It doesn't take very long to set up. So as long as the molecule nanodisc is stable enough for a certain period of time, you can still perform an assay by diluting down in a lower pH buffer. There will be obviously different constraints for certain different polymers. So. But yeah, it's possible to do something as long as it's short-term and the molecule is stable enough.
So there's a question from Michael here. He's asking, what would the ideal polymer look like for your screens, and which properties are key for detecting fragment binders? So for example, less aromatics or charges on the polymers.
So yeah, the ideal is going to be something which is going to be able to efficiently solubilize and purify a protein and keep it stable. So with any of these biophysical assays, the most important thing is the quality of your protein. Is it happy? Is it stable? So you want something which is monodisperse, clean. So otherwise you're going to start getting false positives and artifacts through from your assay. We haven't done extensively lots of different polymers to see how that affects the binding affinities, or whether or not we can detect different molecules being able to bind. But yeah, so I'd say at the moment the most important thing is just the stability of protein. So whatever you put in terms of protein into the assay will affect what you get back out in terms of results.
Thank you. There's also a question from Oliver. He's wondering whether you think you'd obtain the same results if you express the receptor, for example, in mammalian cells, like HEC293 versus insect cells can use the same polymer.
Yeah, it's actually something we are looking into. We are currently repeating, so this was done with the thermostabilized A2A protein. We're doing the same thing with a wild type in both mammalian XP293 cells and in insect cells. So it's the sort of thing we'd want to use this platform to ask these questions. So are we going to see differences in affinities? So interestingly, the thermostabilized mutant doesn't bind adenosine very well. So this kind of assay setup is not going to be great for looking at agonists. It's going to be fine for looking at antagonists, but so we want to do this with the wild type and see if we can shift or to see a change in affinities with agonist molecules. So there's it's ongoing work that we want to try and build these case studies.
Thank you very much. That's really fascinating. I'd like to thank you and also Claire as well, both of our speakers, for really interesting presentations. And I'm going to hand over to Clarice because I think our next session is starting at 2:30. So keep the questions coming, write them in the chat, and the speakers will answer them as we go along. Thank you.
So thank you, Joanna, and hello everyone. So I'm Clarice. Maybe we can wait like 2 minutes if you want to have a little pause before the second session.
Hi, Clarice. Just wanted to thank you for chairing, and maybe you could just tell us a little bit about your research and about where you're working, because people may not know you so well. Would you be able to say a little bit about your project?
Yes. Well, actually, I'm working on nanodisc also with a membrane protein in it, and I'm working about the dynamics between lipids and membrane protein, and I'm doing this in liquid state NMR, but under high pressure.
Oh, great. Could you say what the biggest challenges are in using polymers or nanodiscs for you?
What do you mean by, I mean, I'm not using, well, I'm using a nanodisc, but not polymers nanodisc.
Okay.
I'm using nanodisc constituted by, scaffold protein.
Sure.
Yeah.
Okay.
So,
Are they working well for you?
yes.
Oh, very good. And do you make your own MSPs or do you purchase them?
Yes, I'm using MSPs.
Okay. Well, great. Thanks so much for sharing. I know this is, I asked you last minute and I really appreciate you helping out. So thanks so much. And I think we could probably begin now if our next speaker is ready.
Yes. So welcome to our second talk session. We are going to hear at the beginning from Francisco Barrera. He is an associate professor at the University of Tennessee, Knoxville, and he's going to talk to us about cholesterol controls, the assembly and activity of the EphA2 receptor.
Hello, how are you doing?
Looks great. Yeah, thanks. Just full screen
Yes.
and you're good to go.
Thank you.
Can you see it now well?
Perfect. Thank you.
All right. Thank you, Clarice, for this nice introduction. So today I'm going to be talking to you about the EphA2 receptor. EphA2 is a human receptor tyrosine kinase, which, as an interesting characteristic, it binds to another membrane protein. So the ligand is attached, in this case, ephrin A1 is attached to a second cell. So the binding between the ligand and the EphA2 receptor effectively links two cells. So as a result, EphA2 is involved in cell adhesion, cell migration, and cell communication. EphA2 is primarily expressed during embryonic development and where it controls the development of neuronal networks. However, in adults, EphA2 is expressed in cancer cells, where it has a very interesting and important role. So in standard conditions, EphA2 is in a monomer-dimer equilibrium, as most receptor tyrosine kinases. The dimer is stabilized by the binding of the ephrin A1 ligand. This has an anti-oncogenic effect because the cells remain happily attached to each other and they don't metastasize. Additionally, these dimers can progress into larger oligomers forming membrane clusters. A second time of EphA2 inhibition does not require the ligand, and in this case, kinases like protein kinase A phosphorylate EphA2 in certain residues, and this paradoxically induces an opposite effect. In this case, EphA2 is not a tumor suppressor, but is an oncogenic protein. In this case, you have the opposite phenotype, and EphA2 leads to metastasis. So for this second ligand-independent mechanism, there's not a clear understanding of what's the oligomeric state, monomer, dimer, or oligomers that lead to this metastatic phenotype. So this is where we came in, and we were not satisfied with current methods to study the oligomeric state of EphA2. So we collaborate with our colleague Rajan Lamishani, and we developed a method that we call simple pop. In this approach, we express EphA2 labeled with GFP into HEC293 cells, and then we form a native-like nanodiscs adding DIPMA. As this audience knows, DIPMA forms DIPMOBs, these lipid nanodiscs extracted from the plasma membrane of this case, HEC293 T cells. So for our approach, what we do is we coat a microscope slide with an antibody against EphA2. Whenever you incubate this sample with DIPMOBs generated from the cells, you're going to have essentially all membrane proteins present. we take advantage of the antibody to incubate all this sample in such a way that only Fe2 GFP is going to be immobilized into the slide. And then we perform a TERF experiment where we see that each of these spots that you can see into this image here represents to a single DIMOB. On the left, you have images for slides that were not coated with the antibody, where you see very little sequence coverage. So what we see are traces like that. We take videos and then we see how each of these spots changes and photo bleaches. So whenever we have a DIMOB with a single copy of Fe2 GFP, we have a single step into photo bleaching. When we have two, we'll have two photo bleaching steps, and so on. So when you analyze hundreds or thousands of these videos, you are able to, after you do a correction for the incomplete maturation of GFP in the cell, you are able to determine the oligomeric state of Fe2, where you can see that in control conditions, in the absence of added ligand, Fe2 is mostly a monomer. However, as expected and reported in this paper by our collaborators Adam Smith and Matthias Buch, you also see oligomers of Fe2, dimers, and larger oligomers, trimers or tetramers. When these experiments are repeated in cells that had been incubated with a ligand, as expected, we see a reduction of the levels of monomers and an increase of the levels of oligomers, where we have 3 or more Fe2 molecules trapped into a single DIMOB. This result shows that simple POP is able to characterize and inform of ligand-dependent oligomerization of Fe2. And thus give us a platform to discover new physiological cues that are able to control the self-assembly of Fe2. So we are interested on the effect of lipids on membrane proteins. So we studied the effect of cholesterol using our assay. To lower the levels of cholesterol in the plasma membrane, we use the established assay of using methyl beta-cyclodextrin. This ring polysaccharide is able to interact with the surface of the cell, the plasma membrane, and extract by encapsulation cholesterol molecules from the cells. We have done this experiment in several cell types. In this case, you have data for HEC293T, and then we see that there is roughly a 30–35% of extraction of total cholesterol. Importantly, in our conditions, and this is not always the case with cyclodextrin, you have to do some careful controls. We showed that the extraction of cholesterol is not associated with a decrease of viability. So once we had extracted cholesterol, we repeated the simple POP assay, and this is the data that I showed you before for control and addition of the ligand. So in greenish blue, you see the data for cells treated with cyclodextrin. So you see a very striking decrease of the levels of monomer and also a large increase of the levels of oligomer. So this really conveys the idea that removing cholesterol has an effect very similar to activation of the receptor. Cholesterol, this data suggests, would be a strong inhibitor of the assembly of Fe2. What happens if you do both treatments? You incubate with FP1FC and remove cholesterol. That's where the purple results are. And you see that the results are very similar to cyclodextrin alone. So this tells you that the receptor really, if the levels of cholesterol change, becomes insensitive to the ligand, and it really gets an activated-like state by just changing the composition of lipids. So we wanted to understand how this could be important functionally. So we studied this serine phosphorylation, which is associated with the oncogenic phenotype of Fe2, and we did these experiments on the HEC293 cells where we do the simple POP, but then we also did it in other cell lines where Fe2 without the GFP is endogenously expressed. Due to the limited time, I'll just talk about the endogenous results, where you can see that, for instance, in A431 cells, there is a significant increase of the levels of serine 897 phosphorylation, this being a telltale residue for oncogenic activation of Fe2. So cholesterol, by inhibiting the self-assembly of Fe2, is also, it seems, holding this oncogenic activation of Fe2 and preventing this uncontrolled activation. So to summarize, so in the presence of normal levels of Fe2, in the absence of ligand, Fe2 is primarily monomeric. However, if you decrease the levels of cholesterol, you see that Fe2 is going to self-assemble. I didn't have time to discuss it here, but we studied the molecular mechanism of this process, and we propose that this does not happen by direct interaction of cholesterol to the transmembrane domain of Fe2, but by cholesterol activating beta-adrenergic receptors, which in turn increase the levels of cyclic AMP, which activate protein kinase A. And it's this kinase which acts on Fe2, induces a conformational change on the intracellular domain that then facilitates the self-assembly of Fe2. And we think that this is how cholesterol acts as an inhibitor of the self-assembly and activity of Fe2. I would like to thank you for your attention, and especially my group, particularly Ryan Shack and Alisa Ward and Diane Santana, and collaborators at the University of Tennessee, Rajal Namishani, Jos Vasile, and Ioannis Sgouralis, and also Matthias Bach and Adam Smith that participated in this project and funding to NIH. Thank you.
Well, thanks, Francisco, for this very nice talk. There is a question from Michael in the chat, and it's linked to my question. He was asking how feel it to test other lipids effects, for example, phosphoinositide. Is it feasible? And my question was more like, why did you use the cholesterol at the beginning? How did you think about this one and not another lipid?
Yes, good question. So we published a few years back a study where, in this case, we didn't use the full-length receptor. We work on a membrane fragment of the receptor, and then we showed that PIP2 promotes the self-assembly of Fe2. And then we also have other lines of investigation dealing with other lipids, but certainly these are two very interesting lipids, but there are other very important lipids that we are interested in studying.
Okay, so it's a project to maybe use all those lipids to see the effect on the.
Yes, well, cholesterol it's probably an obvious target because cholesterol really binds or modulates the activity of really many membrane proteins, but that was the lowest hanging fruit, that we'll investigate more lipids.
Okay. We have another question from Oliver. Can you also change cholesterol content in SMARTs?
Good question. I'm sure you can. You could decrease it with the cyclodextrin. In this case, we chose to do it into the cell, but certainly I think that you can decrease it and also increase it using cyclodextrin into the nanodiscs.
Well, thank you. Feel free to ask other questions to Francisco in the chat, and he can answer. For the second talk, we are going to listen to Frank Bernhard. He is a professor at the Goethe University Frankfurt, and he's gonna talk to us about Cryo-EM structures of cell-free synthesized GPCR G protein complex in nanodisc. So Frank, if you want to share your slides, your screen, thank you. Frank, we are not hearing you. Maybe you can unmute your micro.
Forget. So again, thanks for the introduction. And yeah, first, a correction, I'm not a professor, I'm PI, but at a different position at the Goethe University. So I would like to introduce you into two topics. First, the generation of Cryo-EM samples of GPCR G-protein complexes synthesized by cell-free expression. And
Craig,
second,
do full screen, please.
Yeah. And second,
Perfect.
the transfer of your in vitro or self-free synthesized GPCRs back into living cells. And both topics are based on nanodiscs. So that is a scheme how we synthesize our samples. So it's based on we have an in-house cell-free expression platform based on E. coli lysates, and we synthesize the GPCRs or other membrane proteins co-translationally into preformed nanodiscs. These nanodiscs we assemble with different lipids or lipid combinations, and we first check which lipid or lipid combination fits best the given GPCR or membrane protein target. In addition to the nanodisc, we add purified G proteins, agonists, and if possible or necessary, stabilizing nanobodies or antibodies into the cell-free reaction. And you see it's a modular system, so if you exchange individual components, you can easily adjust the system to a new other target. So the whole procedure is completely detergent-free, solves a lot of problems, and most important is that in contrast to many traditional cell-based systems, which have to synthesize GPCRs heavily engineered, so with truncated terminal loops or truncated internal loop 3, or with the addition of stabilizing domains, we only synthesize the GPCRs as pure full-length protein, and they are only modified by a terminal purification tag, strep tag or his tag. So this is a flowchart for Cryo-EM sample production. So you set up the reaction in the afternoon, you incubate overnight, next morning you make a purification. And then in the afternoon, you're already ready to load your grids. So in the two examples I show you, was a histamine 2 receptor and the beta-1 adrenergic receptor. Out of 2 milliliter of cell-free reaction, you end up with finally about 30 microliter of sample, which is good enough for 8 grids. The nanodiscs have a good advantage that they do not have any preferred orientation on the grid, so you can collect sufficient classes for structure calculation. So in case of the human histamine 2 receptor, we came down to a resolution of 3.4 angstrom, which is in good comparison to resolutions of similar structures here of the histamine 1 receptor with GQ that has been published previously. In addition, if you compare now our structure with all histamine receptor structures that have been published before, you see again, all of them contained significant deletions or even insertions of stabilizing domains. So our structure is the first full-length structure of a histamine receptor, and it was also the first of the histamine 2 receptor with GS in coupled to GS in nanodiscs. Certainly some interesting molecular details. I just show you one. So for ligand bindings, this residue at TM5 was always suspected or proposed to interact with histamine. Histamine is a common ligand for all 4 histamine receptors. We could show that in the histamine 2 receptors, TM5 is not involved in ligand coordination. However, this residue is essential for the interaction with histamine 2 receptor specific ligands or agonists. So this is kind of a selection residue for the histamine 2 receptor. Second example I want to show you is the human beta-1 adrenergic receptor. The beta-1 adrenergic receptor is among the best studied GPCR at all. I guess in more than 20 structures have been published, but all but one of them are from Turkey and not from human. And the reason is that the human GPCR, the human beta-1 adrenergic receptor, is highly sensitive against detergent or detergent extraction. And all structures that have been published before are heavily engineered, contains the truncated N and C-terminal ends and also truncated internal loop 3. So this is the first full-length structure of a beta-1 adrenergic receptor, and it's also the first human beta-1 coupled to GS. The additional domains, the terminal domains and the ECL3 are not structured because they are highly flexible, but they have constraints to the adjacent areas. You see this already here in TM5, which is significantly longer if compared to the truncated version. And if we have a closer look at the interface here of the GPCR with G alpha and G beta, we also see significant differences. So here is a full-length interface, and the white lines are potential polar contacts up to 3 angstrom. And you see that the full-length receptor makes significant more contacts if compared to the truncated version. In particular here, the elongated TM5 has additional clusters interacting with G alpha, and we even see clusters of interaction of the GPCR with G beta, and all these are not present with the truncated version. So this shows that indeed, it's a difference whether you analyze a full-length or a truncated version. Finally, I would like to show you another topic. Besides making structures, you're certainly interested to analyze the function of your cell-free synthesized GPCR or membrane protein. And this is a very new technique to bring this in vitro synthesized membrane protein back into the context of living cells. You simply incubate both together, and even within minutes, you see a transfer from the nanodisc into the cell membrane of your GPCR. Also, the lipids are transferred, and the scaffold protein is going off. The transfer is continuing, so you can modulate the amount of transferred protein just by incubation time. And the GPCR, you see here, GPCR MNG fusion is evenly distributed among the cell population. This also is in contrast to traditional transfection, where you always have some cells producing the GPCR like hell, while others don't produce at all. One thing I also want to show you is that by this technique, you can also address or modify primary cells. So here on the right, you see a fragilely isolated heart muscle cell, and those cells are not accessible by transfection because you simply cannot cultivate them. There are certainly other very interesting applications. I just show you one, and this is a dimerization of our cell-free synthesized GPCR with GPCRs which have been synthesized inside the cell after transfection. So what we did is, we transfected templates for GPCRs labeled with a flag tag, and we made our cell-free synthesized GPCRs in nanodiscs, and they are labeled via strap tag. And so first we transfected cells with a histamine, with a flag tag labeled histamine 1 receptor, histamine 2 receptor. And then we transferred self-resynthesized histamine 1 and histamine 2 receptors in these transfected cells. After incubation, the cells are lysed, purified by the strep tag that you see here, and then potentially interacting GPCRs are identified by immunoblotting via the flag tag. And this you see here. So you see a pull down, yeah, of the cellular synthesized flag tag histamine 1 receptor by the cell-free synthesized transferred histamine 1 receptor. So here you see the homodimerization. And here you see the pull down of the transfected histamine 1 receptor with a transferred cell-free synthesized histamine 2 receptor. So here you see the heterodimerization. As control, we transferred a free fatty acid receptor, which certainly does not interact with the transfected histamine, with the transferred histamine receptors at all. And this is a vice versa experiment. So this is a very nice technique also to identify cellular interaction partners. Okay, with that, I'm at the end, and I thank my collaboration partners and funding, which have been involved in that project. And I thank you for attention.
Well, thanks, Frank, for this beautiful talk. We have some questions in the chat. The first one is from Michael, who is asking, how feasible is it to generate modified form like polymer-encapsulation in your CF methods?
Yeah, we have an E. coli system. So in the cell-free system, if you do not add the modifying enzymes, you will get the plain GPCR. And this is also a very interesting application of this nanotransfer. So because the transfected GPCRs are certainly modified, and our cell-free transfer GPCR is not modified, so you can check what role those modifications may play. Yeah.
Thank you. We have another question from Joanna. Does the cell-free expression system have only E. coli lysate components, or have you tried supplementing with lipids enriched in mammalian systems as well?
We pre-fill or we pre-assemble the nanodisc with all kinds of different lipids, and those are certainly also mammalian lipids. So most promising are negatively charged lipids. So the two structures I have showed you are made with nanodiscs filled with phosphoglycerols. Certainly you can add lipids like cholesterol, which are or PIP, which are known to be important for the function or maybe also the dimerization of GPCRs into those nanodiscs. So it's completely free and up to you with which lipid or lipid combination you pre-assemble the nanodisc, and then you add it into the E. coli lysate.
We have a last question from Oliver. How large is the fraction of correctly folded receptor in the nanodiscs?
Yeah, that's a very good question and very important at all. This depends on the GPCR. So we have GPCRs which are folded up to 80–90%, and others like the histamine 2 receptors I showed you the structure of. And this one was folded approximately 30 to 40%. But you can nicely screen this by size exclusion chromatography, because those which are not folded are usually also not completely inserted into the nanodiscs. This co-translational insertion is a pure artificial insertion mechanism. It is completely independent of translocons, SEC, or whatever. So it's a pure physical interaction of the nascent polypeptide chain of the GPCR or any other membrane protein with the membrane of the nanodisc. And it also only works with nanodiscs or nanoparticles. It would not work with liposomes, because here's the physical barrier for the for the translocon-independent insertion is far too high. So this is basically a key point here of this technique.
Okay, thank you, Frank, for your answer. Thanks a lot for this nice talk.
You're welcome.
And to finish, we are going to listen to Evelyn Okorafor, working at the Miami University Oxford, and who's going to talk about the charge effect of SMA derivatives on the membrane bilayer using solid-state NMR. Thank you, Evelyn.
Thank you so much. Sorry, 1 minute. Let me just set up my screen, please.
It's good.
Can you see my screen?
Yeah.
All right. Thank you so much for inviting me, and thank you for the opportunity. I'm actually a graduate student, and I'm excited to be the only student presenting today. So first of all, why do we care? We all care. We are all here because we need to study membrane proteins and their functions to human life, including other organisms. And several systems, like I've listened to amazing talks today, several systems have been employed by experts. And while they've been used at different points, we know they all have their own limitations. So depending on the kind of research you're doing, you choose the best polymer or kind of system that works for your membrane proteins. Today, I'll be focusing on styrene malic acid. So first of all, styrene malic acid have been used to solve a lot of membrane protein structures using Cryo-EM, solid-state NMR, and several others, X-ray crystallography. But SMA also has its own limitations. As you can see, at lower pH, less than 6, SMA precipitates. Also, at a concentration above 10 millimolar for divalent cations like magnesium and calcium, it also precipitates. And that's why we introduce SMA derivatives. So for the course of this research, I'll be talking to us about how we use these derivatives on solid-state NMR to be able to get the right environment in case we need to study our membrane proteins with solid-state NMR. So we introduce SMA glue and SMA AE, which are negatively charged. SMA neutral is electroneutral charge, while SMA positive is positively charged. For the materials we'll be using for this particular research, we'll be employing two lipids, POPC and POPG, in a combination, and we're using a 9:1 molar ratio for this study. So because of the instruments that we are using for this study, our hypothesis is that this looks like our control sample, or this is what our control sample should look like. We also call it the powder pattern, and it has a very large bulky bilayer showing here. And this kind of spectra tends to become isotropic upon the addition of polymers in solubilizing them. So our questions today will be, what will be the optimum ratio of our polymer to lipid that we need to turn this bulky bilayer into an isotropic peak that represents our nanodisc? And since we know that the polymers that we'll be employing for this research are charged, we also want to see what these charges, how these charges can affect the chemical shift in our lipid bilayer. So a little bit of the instrument, solid-state NMR is used to check the structural and dynamic properties of our lipids, our peptides, and our membrane proteins in the lipid bilayer. And because our phospholipids have 100% abundance of phosphorus, we are using the 31P nucleus. And to get the kind of spectra we need for solid-state NMR, it is defined by our sigma 22, sigma 11, and sigma 33. This tells us how the pattern of our spectrum should look like. And as you can see from here, the blue peak is an isotropic peak, which is the reduction of width of all these larger peaks. So these larger peaks are like the control samples, and upon addition of maybe your protein or your polymer, it tends to become isotropic, especially when it is the polymer. So at this point of getting the isotropic peak, we believe that there's full solubilization and that our sample has formed nanodisc. And this reduction of width for the chemical shift and isotropy tells us about how fluidic the membrane is and how dynamic our vesicles are, as well as how decreased in size, like I said, forming the nanodisc, our vesicles are when we add membrane proteins or polymers to them. So for my methodology, I'm also using the thin film method of preparing our samples, our lipid samples in a thin film, hydrating them with our buffer and incorporating them into SMA. In this point, we are using SMA derivatives. And first of all, I check on DLS to confirm that they form the required sizes before I go ahead to check what the dynamics of the bilayer will look like on solid state NMR. So for my results, first was my SMA positive, the positively charged polymer. With DLS, we noticed that all of our samples in all the ratios from ratio 1.5 to 1 of polymer to lipid to ratio 3 to 1 of polymer to lipid, they all form nanodisc depending on the sizes. Although the smaller size, the one that had less concentration of polymer, which is this one, had a larger size of nanodisc compared to when we increase the concentration of our polymer. With solid state NMR, we noticed that they all formed isotropic peak close to the 0 ppm, but we saw an extra peak or an extra shoulder upon the formation of all these isotropic peaks. And as you can see, the 2.0 ratio is the one that has less shoulder attached to its peak. So at this point, we tend to suggest that for positive, the 2.0 to 1 ratio is the optimum ratio. So we went ahead to check with SMA-AE, which is net negatively charged, and also our DLS, they formed nanodisc. But like solid state NMR showed us same thing that we saw with SMA positive, that upon the addition of these polymers at increasing concentrations, we noticed an extra peak or a shoulder to our spectra. But with the 2.0, the extra peak was pretty small, and we also saw that with SMA positive and AE, the 2.0 optimum ratio, the 2.0 was the optimum ratio. Sorry, please. Interestingly, we introduced SMA neutral. This one is neutral charged. And with all of the ratios, as you can see, they kept forming nice isotropic peaks without an extra shoulder, as well as DLS gave us good nanodisc for all of our samples and all of the ratios. At this point, we began to think about what reasons could have caused the extra peaks with the charged polymers. So we suspected maybe there was an incomplete solubilization, or probably there was an intermediate structure between the point of being fully solubilized and the point where they started being solubilized. And because we are using two different lipids in a combination, like we're using a mixture of POPC and POPG, and POPG is negatively charged, we are also suspecting that probably there is an interaction between the charge of the lipid and the charge of the polymer, since we are looking at 31P, which tells us about what happens at the lipid head group. So with this, we introduced more polymers with same charges as the one we already used to confirm our final results. Here, the first two peaks are the negatively charged polymers. At this point, aside AE, we introduced SMA glue, which was negatively charged. And as you can see, the two peaks are comparably the same. This is SMA positive, which is positively charged, and it kind of looks alike with the other charged polymers, the AE and the glue. And then we introduced a new polymer, which was net neutral charged, comparing it with our neutral polymer, and they both formed very nice isotropic peaks without an extra shoulder. And here is our control sample, and you can see how broad it was originally. Now with DLS, you would also notice that all of this sample, because we saw that 2.0 was our optimum ratio, we used 2.0 for all of them. So with DLS, you would also notice that they all formed nanodiscs. So at this point, it was clear to us that the charges on this polymer actually had an interaction with the lipid bilayer, which is our POPC and POPG that we use in corresponding to the 31P nuclear in solid state NMR. So we concluded that 2.0 ratio was the optimum for our charged polymers, and that neutral polymers, which is the SMA neutral that we introduced, formed better isotropic peaks and might be a better option for us to choose when studying membrane proteins to avoid electrostatic interactions. Also, someone might begin to ask, what's the difference between DLS and solid-state NMR, since you're looking at the size and how isotropic your peaks are? DLS tells us about the size of this nanodisc, while solid-state NMR will give us a clearer view of what is really happening in the bilayer, especially about the charge interactions. With this, our future direction is to try and see what one lipid alone will look like, probably POPC that is neutral charged, and then maybe POPG that is anionic, and probably do another kind of lipid that is positively charged, just to compare our results and know how this charge really are affected in 31P phosphorus using charged polymers. And probably we'll also try the temperature studies with solid-state NMR and go ahead to see if we can incorporate a membrane protein to see how these things work with the charged SMA derivatives. So with this, I want to thank each and every one of you for listening. I want to thank Small for inviting me to this conference. It's a privilege. And I want to thank my PI, Dr. Lorigan, for the opportunity and his support, and everyone in my lab, including everyone that is funding my project. Thank you.
Thank you, Evelyn, for this beautiful talk. It was very clear. There is some question in the chat, but I have one in the thing that you want to do in the future. You talk about temperature studies. What do you expect to see if you change the temperature?
Okay, so most times when you change the temperature, it tends to reduce the size of your bilayer as well on solid state NMR. So the width of the solid, the chemical shift anisotropy, since it's wide at the control, we've never done it with polymers, but we've done it with membrane proteins. And we always notice that as the temperature is increasing, they tend to form smaller nanodiscs or smaller, like your bilayer just gets smaller. So the width of the chemical shift anisotropy gets to get smaller while you increase the temperature. So that's what we've seen with membrane proteins alone in the bilayer. We want to see how the polymers will also be affected with this temperature.
Okay, thank you. So we have a question from Michael. Could you please clarify the molecular composition and concentration of the ideal 2.0 ratio?
So the 2.0 ratio is weight by weight. It's not a molar ratio with a concentration. It's weight by weight. So for example, if we have like 10 milligram of our 10 microliter of our lipid, we should have like 20 microliters of our 5% polymer. So it's really not a molar ratio. It's volume by volume, sorry. We do weight by weight first and then do volume by volume. So it's not a molar ratio for the concentration.
So we have another question from Michael too. Are your neutral polymers available commercially or collaboratively?
Collaboratively, yes. Commercially, no, currently. So we can actually make it by request, but it's not out there commercially. But the BZ that I introduced was commercially from Cube Biotech, just to confirm that our neutral polymer was giving us the right result.
We have a question from Joanna. She's saying, thanks, Evelyn, fascinating talk.
Thank you.
And she was asking, what temperature did you perform these experiments at? And do you think that will change the optimal ratios?
Okay, so because we're using POPC and POPG, we used room temperature, 25 degrees Celsius. And that's why I'm saying that we might need to increase our temperature to see if it really changes the dynamics when we add these polymers. But it's room temperature that we use for these particular studies because it's POPC, POPG. So maybe if we're incorporating something like DMPC, we might have gone high based on the phase transition.
Yes. So from Ojat Alizadeh Zainabad, nice presentation. Did you consider to check in which ratio you have less polymer around your nanodisc after purification?
Mary, can you go again?
Did you consider to check in which ratio you have less polymer around your nanodisc after purification?
Okay, purification of a nanodisc or membrane proteins, please. Because, so this is just a lipid and polymer sample. The reason I started with a 1.5, we already did a research previously to get the optimum ratio for our traditional styrene malic acid. And we started titrating from 0.5 to about 1.75. And they noticed that 1.5 was the optimum ratio. So the reason I started with 1.5 was because we already knew that 1.5 was the optimum ratio for the real polymer itself. So the derivative should be around that point. We needed to know if we needed to add more concentration of the derivatives to be able to get a better result, or we need to add less. So while I started from 1.5, 0.5 I tried, but it didn't really make any difference. So I stuck to 1.5, and that's why I had from 1.5 up to 3 to get the best. And at 2, I found out that that was like the optimum for us. So for your question, I don't know if I'm answering your question. I don't know if I really understood your question, but I feel at the point of 2.0, we had enough polymers to be able to solubilize our lipids. And that would be what we could use for our membrane proteins as well, if we are using membrane protein for the system. I don't know if that answers your question. You could ask it if the person is online.
Yeah. He said thanks, so I think you answered well.
Thank you so much. Thank you.
We have a last question from Francisco. Have you observed the extra NMR peak at minus 10 ppm with DIMBA or SMA 2000?
No, we didn't try DIMA or SMA 2000. I actually did SMA 221 synthesized, RAF synthesized by our lab as control, and it was okay. Like, it was very, very okay. It showed the same result, similar result like the neutral, but I didn't have to present it here because my time, it's just 8 minutes. I had to run out everything, but I tried 221, which is very similar to the commercial SMA 200 or 2000, and it gave us a similar result to the neutral, which was nice. But because of the limitations we have with these traditional SMA, that's why we're introducing the derivatives. So we need to optimize the derivatives to be sure that when we're using them for solid state NMR, we're not seeing the 2 components that actually appears, and we need to know which of these derivatives works better, at least with solid state NMR, especially when you're looking at a charged membrane protein that might be affected by the charges of these polymers.
Well, thank you, Evelyn, for your very interesting talk, and thanks for the 3 presenters during this second session.
Thank you so much.
Thanks all of us for coming and joining and for the question.
Okay, thanks everybody. Really appreciate all of your help today. And I'll share the video probably later on tonight.
Thank you.
Thank you.
Thank you.